Hello, class. I just recorded this lecture to almost to the last 4 slides, and then the internet cut out. And apparently, I think I need to start at the beginning again. Doesn't concern you. You don't know this, although I just told you, but I'm gonna start again here on part 2 of the first lecture on the nervous system. Yes. All right. What do I know? I'm just going to start again here. Talking about the synapse. And amazingly, unlike inside your television or this refrigerator or anything like that, the wires aren't connected. Imagine you have a wire with electricity going down, and then it stops. And then you have this little gap where chemicals are released. They travel across this little gap, and they go onto receptors on the other end where they bind with it, and then they excite the next one. It's got to take more time, and it does. It slows things down. The wires aren't connected. There's this chemical gap in there. Why do we do it? What's some of the consequences of this? Well, because there's the chemistry involved. Here you can have drugs and chemicals that really modulate your nervous system, right? You can have amphetamines, cocaine, things like that get you all hyped up, and you have depressants like alcohol and other depressants that slow things down. Indeed, and that's how a lot of drugs work, by working at the synapse. You can have drugs that compete with a neurotransmitter. They can block up the receptors. They can fool around with the recycling of the neurotransmitter so it stays there longer. So all these drugs, can modulate your moods and your nervous system. And so that's one thing. Another thing is it's one way. Because we talk about this in the beginning, the first neuron being the presynaptic, then you have the synapse, that synaptic cleft, a little space, then the postsynaptic neuron. And it can only go one direction, the signal. It can only go one direction. So it's got that too going on. Yeah. And these little red dots are the neurotransmitter. You guys remember what that was in the neuromuscular junction we just did with the muscular system? Acetylcholine, that's in fact true. And we'll talk more about the synapse here, but the chemical is released into this cleft, it travels across and binds to receptors. Yeah. And the amazing thing that I didn't really get when I first was studying this is that one particular neuron just doesn't talk to another neuron. It's the one neuron in question can have 10,000 other neurons that are attached to it. And that neuron itself can make a decision whether to fire or not based on all the information it gets. So some of these neurons will be excitatory, like, what's that black? Is it a bear? I see this black subject over there, and I'm in the woods, right? And the other half can be inhibitory, like, it's a black rock. I swear it's a black rock. After saying it's a bear, should you run or not? Well, should you run anyway if it's a bear? But that's not the point right now. And so some of these neurons will come from your memory, like, oh, wait a minute, I've seen a bear before. It's the wrong shape. Or your mind will say, no, it's February. Bears are hibernating now. I think it's telling you no. But other ones will say, that's fur. It looks like a bear. You should run. So anyway, I'm going on this analogy way too far, but that's the idea of how complicated your brain is, and that it can sum up all these signals, whether to fire or not. Oh, so cool. All right, well, turns out that the wires, if I look at the wires in your TV, they're insulated. They've got insulation on them. And the wires in your body are insulated too, especially the ones you want to go fast. Like the big nerve that comes from your foot up to your spinal cord. If you step on a piece of glass, it is time-sensitive information. The quicker you can lift your foot up, the less damage is going to happen to your foot, pushing into that glass. And so those are big axons that are myelinated. Now, myelin is the insulation. It's this fatty substance that wraps around the axon. And because fat and water don't mix, it's a good insulator. And so myelinated axons have this insulation. And we'll talk about the cells that make this insulation. And even the axons that are not myelinated, they still have some insulation, a little bit, not as good. And so here's I'll tell you the cell. The cell's called a SCHWANN cell. And I'm sure you remember that from high school biology. The SCHWANN cell is going to insulate the axons in your peripheral nervous system, in your arms and legs, everything like that. And even the unmyelinated ones, you can see the SCHWANN cell still protects them a little bit, makes a little cradle for them, and it's going to help protect them and clean up the debris. It's going to help out, definitely. And so when we talk about the brain and the spinal cord, we're going to talk about white matter and gray matter, right? And the gray matter, when you see that, is going to be the cell bodies and not myelinated axons. White matter is going to be the wiring, because that myelin is whitish yellowish. So every anatomy book has the nerves colored yellow. And I see it, like your big sciatic nerve right on the back of your leg. It has this yellowish cast to it, So whitish yellowish because of that myelin is kind of fatty and has that look to it. Yeah, so Schwann cells. They are the cells, and they're fascinating. I'll show you how they wrap around an axon, and they just wrap around and wrap around. And that myelin, lots of lipids, is going to be a nice insulator. Because the electricity is caused by sodium and potassium ions, which are in water. And so you make this fatty insulation around it, and it can't, the water won't be there, and there won't be any kind of ions moving in or out. And true, Schwann cells have other important functions. At the very end of the lecture, I'll show how nerves, if they regenerate at all, usually the Schwann cells will make a tube to try to get that axon to grow through it. So they'll try to help with that. So this is electron micrograph showing all the layers, myelin fatty layers that the Schwann cell has wrapped around an axon. I hope you are impressed as I am. That's a lot of layers. All right, well, it turns out if you took the axon and completely insulated it, the signal wouldn't go. Okay, you need to have these gaps, these nodes, so the signal can be transmitted along it. So the Schwann cells just cover a little segment, and there's these nodes of Ranvier. Again, my French is terrible, but these little nodes here are going to be gaps where the axon is naked. And that's where the depolarization or the ions can move between the inside and the outside of the axon. Between the nodes where the Schwann cells are, completely insulated, covered by this fatty coating, nothing can move. And so this takes the signal, instead of having to slowly make its way down the axon, it can jump and goes quicker. And that jumping, or dancing, jumping, saltatory, means jumping. So saltatory locomotion is what rabbits and frogs do. It just means jumping. And so that is going to speed up the travel of the signal if it can just jump between the nodes. It doesn't have to go all the way down. And this just shows beautifully, and I'll show you another picture here, a myelinated axon out in your arms and legs has a Schwann cell wrapped around it, wrapped around it. But even the unmyelinated, and these might be nerves that carry the signal that tell you, oh, my tummy is full, it's not a big rush compared to I stepped on a nail. So your body doesn't over-engineer. And so if it can get away with a cheaper nerve, not as well insulated, it will. Because those babies will be cheaper than the more expensive babies with all the bells and whistles. And so indeed, the more is insulated, the faster the signal can travel. And this shows a micrograph just showing all the A's are axons. Yeah. So it's showing one Schwann cell is serving many axons in the unmyelinated fibers. So even though they're unmyelinated, they still get a Schwann cell helping out. It's just not well insulated. All right, let's talk about a disease. MS. You've all heard of multiple sclerosis, MS. And it is a disease that is autoimmune, as we've talked about some other ones, that your body is attacking itself. and you make antibodies that are going to attack, and T cells are going to attack myelin. Can you imagine attacking myelin in your body? It's so dumb because that's the insulator of your nerves, you know. And MS is what happens in the central nervous system. So it happens in your brain and spinal cord. When the myelin is attacked, it gets inflamed, and then it just turns into scar tissue. And that's what a sclerosis is. It turns hard or scarred. And this disease is kind of hard to diagnose. Many other things could cause, an onset of vision issues because your optic nerve is often first affected, and then your arms and legs, your periphery is weakness. And so it can be many things can cause this. So MS, you really need to kind of take scans months apart to show that your myelin is being attacked, and then it's MS. And what can you do about it? There is no cure for MS. It's autoimmune disease. Your body's attacking it. You can slow stuff down by immunosuppressive drugs like steroids, where you tell your body to relax. Don't make as many, don't fight off infection. It should be, but don't attack yourself as much too. And that'll help a little bit. It'll slow things down. But it's a disease. Usually comes about in your 20s normally. A little more women than men. What causes this autoimmune disease? The quick answer is we don't know. We don't know the cause of it. Interestingly, if you look at the how it occurs, a lot of diseases, and I have a slide next talking about epidemiology, how we study disease. A lot of diseases, they center in the equator. There's lots of tropical diseases and cholera and malaria, things like that. But this is one that is more common as you go farther north and south, more north though. And so some people are looking at vitamin D as being an influencer. Slight genetic components, but I believe it's probably an infection, a viral infection. Your body makes antibodies against that fatty coat on the virus, and then it backfires, and it's similar enough to myelin that those antibodies will get into the brain, and then they'll attack your myelin. So that's an idea. And the fact that it's, that is geographically in different areas more likely would support, viral hypothesis, but could also be other things in the environment too, pollutants and toxins. So something living in more, richer countries, I guess I have to say more, some of them are advanced, but the countries that it's possible there's things in the environment that are make you more MS more susceptible, more common. But another thing is, tooth fillings were blamed at one point. We think it's a relatively newish disease too. We don't see evidence of it way back when of this MS. So pretty fascinating. And here it is. I can't be remiss if I didn't tell the word epidemiology, because we're talking about AMP, we're talking about many diseases, but this is studying the pattern and causes of disease. And now with COVID-19, we're looking at, almost daily, you can look at case numbers and deaths, things like that. But for instance, I want to look at this, and I just want you to think real quickly about this. This is a map showing rates of cancer deaths. So this is how many of men and women that per 100,000 that die. Doesn't give me a timeline, but And when you look at this, what's the first thing that comes to mind? Why are you much more likely to die of cancer in Arkansas and Tennessee than you are in Montana or Wyoming? I don't know what you're thinking, honestly. One thing you could come to mind is maybe there's something environmental. There's more nuclear power plants there, or coal-fired power plants with toxins that cause cancer. Possibility. One thing I always want you to keep in mind is healthcare, access to healthcare. And it's also very possible that someone living in Mississippi wouldn't have access to healthcare like they do in Colorado. And that means these are deaths due to cancer. And so the person in Colorado would have regular checkups and they would catch cancer early, whereas someone in Mississippi would go to the doctor once it's more progressed, once they go to the emergency room, they wouldn't have these cancer screenings because these are deaths, right? And so, not cases. So it's possible that you're more likely to die because you don't have good health care and you're poor. And you're going to always catch it later than a richer person that goes to the doctor once a year and gets all these screenings that they can afford. Yeah, that's one thing. Good stuff. And when you look at cancer too, you have to look at what are we looking at here? We're looking at deaths from all cancers, but looking at cancer rates, you can look at this as new cancer cases. You can see breast cancer is way up there, but you're less likely to die when you look at deaths than if you get lung cancer, which is rarer but more deadly. And so all these things play into epidemiology, and I'm fascinated by it myself, although I'm just an amateur. All right. Here's a test question for you, almost guaranteed. I don't know, I guarantee it. Look at the neuroglial and the central nervous system. So remember, brain and spinal cord. We talked about SCHWANN cells in the periphery. They're going to insulate your axons. Let's get into the brain and spinal cord. And here they are. There's 4 different kinds. And I'll show you pictures and I'll talk about all of them. But the SCHWANN cells of your brain and spinal cord are called oligodendrocytes. Oligo means few, and that means one SCHWANN cell in a myelinated fiber is just going to be faithful to one axon. It's going to wrap around it. Oligodendrocytes, you look in the brain, one oligodendrocytes takes care of several axons, or a few of them. Yep. Astrocytes are shaped like a star, like astrology, right? Astrophysics. And these are the biggest, most common ones. They're going to make the scaffolding, and they make scars in your brain you need it, and they're going to cover the whole brain. So those are real common. Microglial cells are the phagocytes. They're going to come from your bone marrow originally, but they make it to the brain, and they're there to eat the bad guys. And the epidermal cells, I'll talk real briefly, they're going to line the hollow parts of your brain that are filled with fluid. All right, so remember, neuroglial cells are the cookie to your chocolate chip cookie. They're 10 times as common as the neurons. And they have lots of functions, supporting functions to the neurons. When you look at brain histology, honestly, the most fascinating part of your body, you could argue that, is the most boring histologically, because all this pink is that's all the axons and dendrites. The neuron cell bodies jump out at you, they're big. And then the other nuclei will be neuroglial cells, but pretty boring tissue when you look at it. And neuroglial cells will be, like I say, the support system to all your neurons. All right, microglial. Normally, if you made a count, only about 5% of them, of the glial cells, will be microglial cells. But importantly, if it's like 20%, that's good evidence you got some kind of brain infection. So they can use that as a marker, definitely. But they're cells that cruise the brain, and they will eat up debris and invaders. Here's a little guy cruising in your brain. All right, astrocytes. They in fact kind of look like a star. They have lots of processes that come out like that. And they really fill up your brain. And as I mentioned, neurons don't divide really, very rarely. And so if you have a big brain injury, the astrocytes just fill in that space with a big scar. It's useless in terms of brain tissue, but they're going to make kind of that scar tissue. Really cool is that the astrocytes are going to make what's called the blood-brain barrier, which I'll talk about in several slides here. But your brain is very babied. It's not in contact with the blood like the rest of your tissues. It has a barrier. And the astrocytes decide what substances come from the blood into the brain. All right, they're the gatekeepers right there. And so we'll see how the astrocytes, the little feet, are going to make this blood-brain barrier. So beautiful, looking at this, you can see it looks like a star because of all these little processes in the astrocyte. All right, so this blood-brain barrier, if you look at, here's a capillary, and if I looked at my muscle cell anywhere else, the capillaries are damn leaky. Stuff comes in and out like nothing, right? But your brain is really protected. So toxins and high sugar levels, the brain isn't exposed directly to those. You've got a little area of protection, this blood-brain barrier. And so if you look at this capillary, this shows some spaces, but realize another astrocyte will come in and will fill up. So there's no bare areas on the capillaries. There's no area where the capillary is leaky onto the brain, because astrocytes will put their little foot processes right next to each other and completely surround it. And they'll be able to shuttle substances, to the neurons back and forth and get rid of waste. So the astrocytes are like the bouncers or the doormen, that are in control of what comes in and out. And here's another view. This one's kind of hard to see, but I like it a lot. This shows a blood vessel coming down, and then it's coming at you down there. See that? So if you look at that capillary coming at you, look at that. It's surrounded by the feet of astrocytes. And you see the astrocytes completely surround it, and they surround the whole surface of your brain too. Their little feet make this whole big barrier. Yeah. And in terms, I know I got some pharmacy people out there, future pharmacists. Drugs, any kind of drugs that you want to act on the brain, you got to design them to get past the blood-brain barrier. And a famous example is antihistamines. The old versions made you drowsy, and then they come up with these anti-drowsy ones. And all they did is they tweaked the chemistry of it so that that substance can't cross the blood-brain barrier, or does it? And and then it's not going to make you drowsy. So it's just one cool example. And last picture, but again, capillaries anywhere in your body are freaking leaky. There's holes everywhere and stuff comes in and out. Another thing, there's a blood testes barrier too. It kind of protects your stem cells and you're making sperm, And then here in the brain, you can see you've got that blood-brain barrier made by the feet of the astrocytes. All right. Oligodendrocytes are the SCHWANN cells of your central nervous system. So SCHWANN cells are out in your arms and your legs and your periphery, and oligodendrocytes are what you find in your brain and spinal cord. As you can see, they don't just do one axon. You can see this one is doing 3 different ones, and they'll wrap around it and insulate it. Yep. So the SCHWANN cell, oligodendrocyte, doing the same thing, the peripheral and the central nervous system. Epidermal cells, your brain starts as a hollow tube. It's hollow. On the inside, it's hollow, and it's filled with this watery cerebral spinal fluid that's going to protect it. It goes around your head. So your brain's kind of in this water bed. It's kind of protected in this water. And epidermal cells will line it. They'll be usually columnar cells. Some of them have cilia, and they're going to line the ventricles. The spaces in your brain are called ventricles. Just like the ventricles of your heart are the spaces. The ventricles of your brain are the hollow middle of your brain that's filled with fluid. Here's some. Enjoy. All right, so what makes the cerebrospinal fluid? We'll get into that for sure, but I guess I'll introduce now the term choroid plexus. And so within the ventricles of your brain, you have these balls of capillaries, and then they're surrounded by these epithelial cells that are making a decision. What do we want to include in the cerebrospinal fluid, this fluid inside your brain? That's when you do a spinal tap too. You take fluid out of your spinal cord, same fluid. So it's watery. It's not just water, but it's going to have potassium and magnesium and things in it. So it's a thin, watery fluid that's made by the choroid plexus, which are these capillary beds that'll be in the ventricles or spaces in your brain. And we'll talk a lot more about it. All right, this is kind of a review picture. It shows the 4 different kinds of neuroglial cells in the central nervous system. Take a look at them. You can see the neuron in the middle, the boss, the star of the show, is a big neuron. You can see it has little processes coming out that are the oligodendrocytes are going to insulate. The most common one of these big astrocytes, you see them, they're making the blood-brain barrier. Their feet processes are protecting and covering everything. Epidermal cells are going to be the cells that surround the spaces in your brain that are filled with fluid. And lastly, microglial cells are like 5%. They're cruising around looking for bad guys. All right, got it? 4 kinds of main kinds of neuroglial or helper cells in the brain besides the neurons. Yep. And here's some words that kind of describe them. You can run your eyes over them. All right. Well, if you remember from biology class, do you remember there are 4 characteristics of chordates? God, I wish I was in class and I could pick on you guys to see if you remember this basic thing from biology. Chordates, vertebrates. One of them is having a dorsal hollow nerve cord. Please tell me some of you out there are like, oh yeah, dorsal hollow nerve cord, pharyngeal slits, postanal tail, right? Well, we all start out, if you look at us, as a ball of cells, then a hollow ball of cells. Then we get this streak on us. And then that, it turns out it's just like a groove, and that groove will get deeper. And eventually you'll end up with a hollow nerve cord along our back. And that nerve cord will become our brain and spinal cord. And the anterior end will get huge and fold and become a brain, and the rest of it will become the spinal cord. But we always retain that hollowness in the middle. It's always hollow in the middle. Unlike a lobster or a bug or something like that, it's solid. So that dorsal hollow nerve cord is a characteristic of us vertebrates, right? Yeah. Indeed. And I don't think I need to go into too much. I don't need to talk about neural crest cells, but early developments, you should know, we begin as a streak that becomes the brain and spinal cord. And I have a good friend teaches developmental biology, and fascinating. You can take a fertilized chicken egg and you can cut the shell away and then put some saran wrap, and you can incubate it and follow that streak, and it develops into a nerve cord, and you see the vertebrae develop and you see the heart start beating. But the ability to look into an egg, like it's hard to do with humans because it's inside of us, but with a shelled egg, you can actually take that shell and carefully peel it off. You leave that little membrane. Yeah, that's really cool. All right. So the glial cells, they do turn over, like neurons really don't, but the helper cells will. And if there's a scar, the astrocytes will divide like crazy and fill that up. All right. So the 4 types of neuroglial cells in the central nervous system, and then we have 2 types in the peripheral. And main thing is SCHWANN cells. SCHWANN cells are the cells that are going to insulate the axons in our body outside of our brain and spinal cord. And then remember, we have ganglia. Ganglia will have a whole bunch of nerve cell bodies outside the central nervous system, and like these little balls that we find in our other places in our body. And then we call these little satellite cells little glial cells that'll be in the ganglia that kind of surround them. So just to be complete, the neuroglial cells of the peripheral nervous system, the SCHWANN cells, and then in the ganglia, you'll have satellite cells that surround the neurons there, and they help protect them and nourish them. All right, and finally, we get to repair of nerves. And you all know one of the injuries you most fear is a spinal cord injury, right? You know that bones, they heal beautifully. I say that. I know it sucks to have a broken bone, but they heal almost good as new, right? I told you a skeletal muscle doesn't heal that well because there are some stem cells, but they don't really divide. Smooth muscle does divide. Your uterus can get big and small. Smooth muscle can divide easily, but and then your nervous system, that the neurons don't really divide. And that if nerves are cut, they can regenerate. And so if you have a severed finger, they can put it back, but you got to put those nerves right next to each other in order for them. And it's time sensitive in order to reattach. If they're too far apart, they both die, the muscle dies. And if a muscle is not attached to a nerve, it atrophies and dies. You got to have that connection. So let me tell you what happens. What happens is that if you have it doesn't have to be even a cut of the nerve. It could be a crushing injury, something like that, where the axon is really, really damaged. The problem is everything beyond that is going to wither and die, because you remember a lot of the substances are produced in the cell body and they go down the axon. So once it's no longer connected, it's all going to die. And if you look at the cell body at the top, you can see if it doesn't regenerate, then the neuron cell body just starts degenerating and it will die. And then the muscle that is attached to, if it's attached to a muscle, that will atrophy away. So here's another good view of it. You can see here, everything's working good. You can see the neuron looks good. The axon is great. The muscle fiber looks great. But imagine you sever it. Well, what happens is that the Schwann cells will try to guide the axon back to where it belongs. And so the axon will branch many places. And if one of those branches hits where the Schwann cells are trying to guide them, it can reconnect. And then look, it's reconnected. The muscle looks fine. But if those things are too far apart, what happens is that the neuron will die, the muscle will atrophy, and will never reconnect. And so a spinal cord injury, if sometimes it can not even be a severing, but just a crushing blow. And the inflammation it's very unpredictable. If everything comes back together again, you're going to regain function. But the fear is that if enough damage is done, and it's very time sensitive, you really have to wait and see. If they don't connect, you'd be, quadriplegic, paraplegic. We'll talk about that. If they don't connect, you're going to lose that connection between the cell body and where it connects. All right. Yes, and this, I kind of like this little drawing here. It shows, imagine a damage to the spinal cord, and you're going to have, when it's severe damage, you're going to have macrophages, you're going to have inflammation, all this happens. And then you have a glial scar tissue, which is useless for conducting sensory and motor stuff, right? So you don't want it to get to be this scar tissue in your spinal cord. You want it to reconnect. And so a lot of research going into, what can we do after a spinal cord injury and even after paralyzation? Is there anything we can do to make that reconnection? And it's a tough job, especially if all those axons have turned to scar tissue. Then I can't imagine how you can regrow that connection. All right, the synapse. We'll talk about the synapse. Yeah, talk about a little bit right now. So this is the cool chemical part of the signal. And so coming down the axon, it's electrical. And we'll talk about what makes that electricity. Electrical goes to the very ends. And then the electricity hits the synapse, and then it's going to cause the release of neurotransmitter into this little cleft or this little gap. And it's going to cross the gap, and there's going to be receptors on the other end for that neurotransmitter. And we call it the presynaptic and the postsynaptic neuron. It's pretty cool, yeah. There's electricity. See those little green spheres in there? They're filled with a neurotransmitter. I'm going to ask you guys, what is the neurotransmitter we see in the neuromuscular junction? Acetylcholine. Yep. And then we'll see there's hundreds of neurotransmitters in your brain. So indeed, this is what I want you to know. We'll talk about the electricity, how that action potential, remember how it's going to reach the end. And then it's going to open up calcium channels. Calcium will rush into that synaptic knob, the end of the axon. And that calcium rushing in is going to cause these vesicles or sacs filled with the neurotransmitter to empty into this cleft, into this narrow space. And that neurotransmitter, be it acetylcholine or whatever, is going to diffuse across, and there will be receptors on the other end for it. And enough neurotransmitter binds to those receptors, it's going to open up channels that are going to excite that postsynaptic neuron and make it fire. At least it's going to excite it. We'll see if it makes it fire. This is the key. Some of these neurons are excitatory, green light, and they're going to excite, depolarize. They're going to I don't get into this quite yet, but if they're going to excite this neuron, others will be inhibitory, and they're going to say, whoa, don't get excited. And so one neuron can have thousands of synapses, and it's going to sum up, it's going to add them up, all the pluses and minuses to decide whether to fire or not. That's what I talked about, example of, is it a bear, is it not, should I run, should I not? It's going to come from all this information that can come to one particular neuron. And that's in some of these neurons for higher thought. In that simple like reflex when you hit your knee, there's none of that. It's just a reflex. But we're talking about other neurons can have thousands of synapses, so they can make some complex decisions. And so if you see excitatory postsynaptic potential or inhibitory, if you see these on a test, E is excitatory and inhibitory is going to slow things down. And so I love this drawing. It just shows so many synapses. And so they'll be in the dendrites and the body. And actually some of them closer to this axon hillock, they have a louder voice than some that are farther away. So there's subtleties like that. But this neuron in question is going to sum, it's called summation, all the inhibitory and excitatory impulses to decide whether to fire or not. Yeah. And if it gets like equal numbers of excitatory and inhibitory, nothing happens. You got to have more excitatory, it's going to reach a threshold and it's going to fire, all or none. So that's what this neuron is making a decision based on all this input that it gets. It's all chemical. So it's not like a real decision, but that's pretty cool. All right. All right, I made it through. I gave this lecture twice, this one. Let me tell you, I'm going to finish this up, but if your internet goes out, your lecture just stops and I don't know where it goes. So, all right, you guys, I hope you enjoyed the first lecture on the nervous system. Again, complex part of A&P. I've got a lot to talk about. In person, I will continue this story and I'll give you more online lectures and you guys read the chapter and I wish you all a good night.